Okay, so let's continue with the cytoplasm and nucleus. In cytoplasm means it consists of all the cellular contents between the plasma membrane and the nucleus. So which is floating in the this yellow part, that is cytoplasm, and it is having cytosol as well as organelles. That is, some of the cell organelles are the cytosol is the cytosol means it is a fluid portion of cytoplasm having water, dissolved solids and suspended particles. Organelles means little organs present within the cytoplasm. For example, mitochondria, Golgi bodies, etc. Then nucleus, large organelle. This is the largest organelle within the cell that house most of cell's DNA. So the nucleus carries DNA, and inside the DNA there is what we are able to see, chromosomes. And from there, this our cancer is getting original. Okay, let's see what is happening. So within the nucleus are some chromosomes present, each of which consists of a single molecule of DNA associated with several proteins. Basically, DNA, the basic unit is protein only. Okay. And a chromosome contains thousands of hereditary units called genes. This is the basic unit of DNA that control most aspects of cellular structure and function. So I hope you are able to understand the basic structural units of the cell, etc. Now let's move on to the basics of cell division, how the cell is being divided. So before that, I will be explaining cancer. Cancer means over multiplication, in our normal words, over multiplication of the cells. That is, more cells are present. Okay. So just imagine there is something is fishy regarding this division process of the cells. Okay. So the cell division have to be we have to thoroughly know what is basically happening, and the changes or the problem is happening for cancerous cells. Okay. The cell division, basically, as cells become damaged, diseased or worn out, they are replaced by cell division. Okay, and not only by this, as a part of growth of tissues, also cells are getting divided and multiplied by cell division process. So basically, there are two type of cell division. This is just for your understanding. You have to understand basically what is happening in our body. We are having somatic cell division as well as reproductive cell division. Somatic cell division means the normal cells other than the normal cells, like for example kidney, the cells within the kidney, stomach, etc. That is somatic cells. So here it undergoes nuclear division called mitosis and cytoplasmic division, that is cytokinesis. Basically, from one cell, two cells are being formed. That is somatic cell division. That process is known as mitosis, that is division of nucleus. Along with this, the same cytoplasm should be there. So that division is known as cytokinesis. Okay, thereby it produces two identical daughter cells. Two identical daughter cells. Now we are having reproductive cell division. It means mechanism that produces gametes, that is sperm and oocytes. Okay. So here it consists of special two-step division called meiosis. Meiosis. So meiosis, which means number of chromosomes in the nucleus is reduced by half. Okay. So the half number of chromosomes will be present in one cell, one product cell. And because in sperm there will be half, as well as the ova will be having half. While they get to united to form zygote, it will be coming into a pair. That is, thereby the reproduction is being done. So then only the entire processes will be happening. So this is regarding the reproductive cell division. This is based on meiosis, while in somatic cell division, this is by mitosis. Okay, but in this reproductive cell division also followed by meiosis, there will be cytokinesis, because we have to divide the cytoplasm also in order to have a complete cell. Then cell cycle, based on this, this division is being carried out. Okay, cell cycle means this is a picture of cell cycle, and you can see some of the write-ups, words, etc. is being there. don't get worried regarding this kind of complicated pictures and all. This is very easy. Let's see. It is an orderly sequence of events by which a somatic cell duplicates its content and divide into two. When a cell reproduces, it must replicate all its chromosomes, so the genes may be passes on to the next generation of cells. That is, we could able to get the same cell as like the parent. Then only the division will be complete. Okay. So the cell cycle consists of major two periods. Two phases are there for basic major two phases are there. That is interphase as well as mitotic phase. In interphase, here the cell is not dividing. That is interphase. But in case of mitotic phase, the cell is being dividing. Okay. In interphase, no division is there, but in mitotic phase, division is there. So in interphase, there is basically three phases. Three phases are including in interphase. That is G1, S and G2. G1, S and G2. Here S stand for synthesis of DNA. S for synthesis of DNA. And G phases are periods when there is no activity related to DNA duplication. Okay, so we can call it as gap. Gaps in DNA duplication, that is G for gap. Here no division is being taken place. So the first phase is G1. Okay, in G1, it is the interval between the mitotic phase and the S phase. Between mitotic phase and the S phase. So here what will be happening? While going through this cell cycle, you have to keeping in mind regarding a cell. Just have a picture of cell in your mind. Okay. So the cell is going to a G1 phase. So what will happen? Here, the cell is metabolically active. Cell is metabolically active and it duplicates most of its organelles and cytosolic components but not its DNA. So DNA is present inside the nucleus, right? Okay, so in this G1 phase, the division of cytoplasmic organelles will be happening. Okay, but no DNA division. That is, nothing will be happen to nucleus, only the organelles. Okay. For a cell with a total cycle time of 24 hours, G1 lasts 8 to 10 hours. So the G1 phase is having the time duration 8 to 10 hours. Okay. Now this is the picture. So let's see the G1 phase. Okay. So the G1 phase, the cell metabolically active, duplicates organelles and systolic components, and the time is 8 to 10 hours. So from here, the division is the cycle is being starting. This is the phase between S phase and mitotic phase. Okay. Now the S phase. S phase means here. S phase. So from the word itself, it is the interval between G1 and G2 lasts about 8 hours. Here the DNA replication occur. DNA synthesis will be happening. So the two daughter cells formed during cell division will have identical genetic material. So when this S phase occurring means two daughter cells will be we will get two daughter cells having same genetic material. Okay, two identical same cells with two identical cells with two same genetic material. Okay. Now we are having G2 phase. G2 phase means interval between the S phase and the mitotic phase. S phase and the mitotic phase. So here what will happen? It lasts 4 to 6 hours, and here during this G2 phase, cell growth continues and enzymes and other proteins are being synthesized. Here the enzymes as well as other proteins, enzymes and other proteins will be synthesized. So basically this protein means these are for having the energy for this mitotic division. Okay. So herewith we will be completing the G2 phase. Now one more phase is there, that is G0. This is the stagnant phase. Here nothing is being happened to our cell. That is, the cell remain in G1 for a very long time, perhaps destined never to divide again. Here, when the cell is getting into G0 phase, no further division is being happening. For example, nerve cells. Nerve cells growth is limited. Okay. So no more further division is being happening in this G0 state. So this is regarding the interphase G1, S phase, G2 phase and G0 phase. Okay.